In this video, we're going to take a look at the introduction to metabolic processes. We'll look at cellular respiration basically as an overview, and later on we'll take a look at photosynthesis. Most of the images found in this video will be adapted from our Cognitive Higher Level Biology textbook, so they'll correspond to what you see in your textbook as well. So recall that metabolic pathways consist of enzyme-catalyzed reactions, and remember these pathways are multiple steps. So they are interconnected, there's many stages sometimes. If you take a look at aerobic respiration, there's basically 4 different stages, and each of those stages have several steps, and they're catalyzed by several different enzymes. Some of those steps also can form, or stages can form cycles, and the final products will be the reactant, and so the cycle can return over and over again. And the example would be the Krebs cycle and the Calvin cycle. An example of a linear reaction would be glycolysis or the link reaction or the electron transfer chain. So before we can really proceed further, we should understand what a redox reaction is. You'll learn about redox reactions in chemistry in greater detail, and you'll even learn how to balance redox reactions. We don't have to worry so much in biology. You just need to understand the basic concept. So redox reactions are chemical reactions that involve the transfer of electrons from one substance to another. So they're called oxidation reduction reactions or redox reactions. So if you lose electrons during a redox reaction, you have been oxidized. So a way to remember it is LEO. So loss of electrons is oxidation. The gain of electrons during a redox reaction is called reduction. So I remember that as GER. The gain of electrons is reduction. So the way to remember this concept is Leo the lion goes GER. You lose electrons, it's oxidation. You gain electrons, it's reduction. It's a little hard to see electrons moving in a chemical reaction though, so we're going to look at this even more in depth. So if we take a look here, this is from our cognitive textbook, not our current version, this is from 2020. But if you look here, the thing that gets oxidized is called the reducing agent because it reduces the oxidizing agent. So the thing that gives up the electrons is reducing the thing that's gaining the electrons. And the thing that's going to gain the electrons is oxidizing the thing that's going to, in the end, lose the electrons. So compound A had 2 electrons that it could lose, and it lost it to compound B. So compound A is oxidized, and compound B, because it gained electrons, was reduced. Again, I mentioned before, it's hard to see electrons moving from one side of the reaction to another because we don't normally show those in a balanced chemical equation. Again, in chemistry, you will learn how to trace electrons, but the way you can identify if something's been oxidized or reduced is if, when it loses electrons, it's also losing hydrogen atoms from that substance. And when it loses those electrons and hydrogen, it's subsequently losing energy from that loss. So if you see something on the left side of the equation had more hydrogen than its corresponding molecule on the right, that thing is being oxidized. On the other hand, if you see something being reduced, the molecule on the left-hand side will have fewer hydrogens than the one on the right-hand side. So it's gained electrons, it gained hydrogen, and when that does that, it gains energy from that gain of electrons and hydrogen. So we'll look at some examples, and this will make more sense, I believe. So just a hint, photosynthesis and cellular respiration are examples of redox reactions. So if we look at glucose here, glucose during the reaction will lose its hydrogens to oxygen. So glucose is being oxidized to carbon dioxide. Oxygen is being reduced to water. So glucose loses the electrons and the hydrogens to become carbon dioxide. So that's an example of oxidation. oxygen is gaining the electrons and hydrogens and being reduced to water. So those are the examples of oxidation and reduction. So another term we should be familiar with is phosphorylation. And that's when you're adding a phosphate group to a molecule. And lots of biological molecules have a binding site for phosphate. When a molecule gets an extra phosphate, it makes it unstable and thus more likely to react. So when something gets phosphorylated, it's primed to react to do something. This leads us to adenosine triphosphate. We call it the chemical currency of the cell. It's the molecule that provides energy to other molecules when that third phosphate is released from ATP and given to the other molecule. When that happens, ATP, adenosine triphosphate, becomes adenosine diphosphate, or ADP, which has less energy within it. And so through other reactions, the ADP will gain another phosphate to become ATP, and then it can power another reaction to happen by transferring that phosphate. Some of examples of times when you would undergo phosphorylation or spend your ATP to make something happen would be when you have to move molecules against their concentration gradient. So often those protein channels aren't called channels, they're called pumps, because you're using energy, ATP, to phosphorylate that pump to push the molecules against their concentration gradient. Also, anabolic reactions like protein synthesis, you're taking energy to build things together, to remove the water and join the other two parts together, and then just basic movement or any other energy requiring functions in the cell. So NAD, or NAD plus sometimes you'll see, is nicotinamide adenine dinucleotide. It's known as a coenzyme. So a coenzyme is a molecule that helps an enzyme do its job. It's a molecule that's easily oxidized and reduced, so it acts as an electron and subsequent hydrogen carrier. So we'll see a lot more about NAD plus or NAD and ATP in the following lessons. So in earlier grades, you would have learned about the mitochondrion, and you would have been told that it's the powerhouse of the cell. It's sort of true, but not entirely true, and you'll learn that a little bit later too. But it is a very important molecule in generating energy for the cell, especially when aerobic respiration is occurring. But anaerobic respiration, which does produce energy, does not require the mitochondrion at all. And that's why the powerhouse of the cell is a little bit untrue. The mitochondrion has two membranes. It is a complicated structure. And if you remember from last year learning about the endosymbiotic theory, that the mitochondrion is believed to have been a prokaryotic cell that got absorbed by a eukaryotic cell and became an organelle. And the reason why we believe that is that the mitochondrion actually has circular DNA, just like a prokaryotic cell. And it can reproduce on its own without having to rely on the rest of the cell to reproduce. So, the mitochondrion, two membranes. There's an outer smooth membrane and a highly folded inner membrane, and we'll look at that in a little bit. Those folds on the inner membrane increase surface area dramatically, and those folds are called cristae. There is space in between the folds, and it's literally called the intermembrane space, so the space in between the two membranes. And then the space inside the inner membrane is called the matrix. And a lot of the reactions of cellular respiration happen in the matrix. And here we can see a representation of a drawing of the mitochondrion. I usually draw mine much more 2D, but that's okay. And then this is an electron micrograph of a mitochondrion. So if you see a structure like this, round or oval, with these projections that look like these lines going across like so, those are the folds of the inner membrane, and these are the cristae. So the folds are the cristae. This is the outer membrane, and then all of this in between is the intermembrane space, and the inner part is the matrix. So a couple of other things that we can't see in this diagram, but the circular DNA, We have the intermembrane space, the space between the two membranes. The matrix is the fluid-filled section. Another reason why we know that these used to be prokaryotic cells is the ribosomes are 70S ribosomes. Those are the size of the ribosomes in prokaryotic cells. And then again, there's the outer membrane, the inner membrane, the cristae, and the fluid in the middle of those folds. The center is the matrix. So one prompt that you need to be able to discuss is, how is the structure of the mitochondrion adapted for its function? Mitochondria is actually the plural of one mitochondrion. So let's have a look. So the inner membrane is highly folded into cristae, and that allows for more space for the membrane proteins of the electron transport chain. You don't know what that is yet, but you will. The intermembrane space is small, so the space in between the inner and the outer membrane, it's small, and that allows for a concentration gradient to build up. The watery matrix has all of the enzymes that are needed to let the reactions of aerobic respiration to occur, so the link reaction and the Krebs cycle. And then the ribosomes that are in the matrix are able to build the enzymes that you need for aerobic respiration. And it might be worth comparing aerobic versus anaerobic and where those processes happen now, and we'll come and visit those again when we look at the steps of cellular respiration. And we're just looking at anaerobic and aerobic in humans. In bacteria and yeast cells, it's a different process for anaerobic. So aerobic respiration in humans includes oxygen. Anaerobic, no oxygen. Where it happens? Aerobic occurs both in the cytoplasm, some of the stages, and in the mitochondrion. Anaerobic respiration occurs only in the cytoplasm. Aerobic respiration can break down all respiratory substrates, so carbohydrates, lipids, and proteins, whereas anaerobic respiration can only break down carbohydrates. Aerobic respiration makes a lot of ATP, way more ATP than anaerobic respiration, and we'll look at the actual numbers later. And then the waste products of aerobic respiration are carbon dioxide and water. And in anaerobic respiration, the waste product is lactate or lactic acid. And we'll visit that again much later. So the overall reactions, the word equations for aerobic respiration is glucose plus oxygen makes carbon dioxide plus water plus ATP. Anaerobic respiration in humans is just glucose breaks apart to lactate and ATP. That's it. Later on, we may be doing some metabolic processes investigations. So you might want to measure respiration rate. The way we do that is using a respirometer. This is a complicated respirometer. If we watch Mr. W's respirometer setup, it's much more simplistic. So you might want to go there and have a look. But what we do is we add potassium hydroxide into the tube right here. And its job is it's going to absorb any of the carbon dioxide that this insect is releasing. So you actually have to put a barrier of some sort to protect the insect so that it doesn't come in contact with the potassium hydroxide because it's an extremely strong base. And then you have this tube. It's a manometer. It's capillary U-tubing containing a colored liquid. And basically, you'll close this tap during the experiment. So there's no more oxygen or no gas can come in and out. So what happens is as the fly or whatever insect this is using up the oxygen and exhaling carbon dioxide, the carbon dioxide is immediately being converted by combining to potassium hydroxide. So you're actually going to create a negative pressure in here. And so you're going to move this tube of water, colored water, so you can actually measure how far this has moved over time. Over here, you might wonder, well, why do they have this? Well, this is just a controlled. So in this example, there's just filter paper and potassium hydroxide. It just absorbs any carbon dioxide that was there, but there's no consumption of oxygen, so it shouldn't move. This is the control.